Hello, my name is Andrew Kendall, and I represent the European Squirrel Initiative, ESI. ESI was formed in 2002 as a charity with the aim of eradicating the grey squirrel, leading to the eventual return of our native red squirrel. Over the last almost 20 years, ESI has looked at various methods to achieve this eradication, and has looked at new science, looked at traditional methods, and various ways of controlling the greys. About 3 years ago, we met with Professor Bruce Whitelaw at the Roslin Institute to look at gene technology as a means of achieving our aim. And working closely with him and the Roslin Institute, we have made enormous progress in the area of gene editing and gene drive. And we now believe that we have come up with a solution that can not only be used to control grey squirrels, but also other invasive species. I'll now hand over to Professor Whitelaw, who can give you a more detailed presentation of gene editing and gene drive and how it can achieve our aims and objectives. Thank you very much.
Thank you, Andrew. And I'm just going to share my screen with everyone at the minute. So I hope you can all see this screen. I'm going to spend a few minutes describing to you the idea which Andrew just alluded to, and that's based around gene drive, a genetic solution to the humane control of animals, pest animals such as the squirrel. But first, I want to explain who I am and my team here in Edinburgh. I'm Bruce Whitelaw. I hold the Chair of Animal Biotechnology at the University of Edinburgh. I am currently the Interim Director of the Roslin Institute. The other face you see on this slide is my colleague, Gus McFarlane, who started the project while being a student in my group, successfully defended his viva a few months ago, and is now Dr. Gus McFarlane. A little bit more about where we are. The Roslin Institute will be known to many of you for a certain sheep called Dolly. We are just currently celebrating 25 years since Dolly and are involved in a number of media activities around that. The Roslin Institute is fully within the University of Edinburgh, one of the top universities in the world, currently ranked 16 on the international scale. We've come some distance since Dolly, utilizing modern day genetic engineering tools, the most exciting of which is called CRISPR, or clustered regularly interspaced short palindromic repeats. These are molecular enzymes that can cut DNA in very precise manner, and we can then engineer the cut site to be repaired in, again, a very precise manner, and thereby change the genetic makeup of an animal. To illustrate this is the picture in the bottom right-hand corner, that of a bunch of pigs we've made here at Roslin. These animals have been engineered using CRISPR technology, genetic engineering technology, to make them resistant to a virus called the porcine reproductive respiratory syndrome virus. For those of you involved in the pig industry, you'll know how important a breakthrough that is. So the idea that we conceived and shared with colleagues in the European Squirrel Initiative, ESI, is captured in the title in the top left-hand corner here, CRISPR-based gene drive for pest control. Since proposing this a few years ago, there has been progress elsewhere in the world to indicate that this concept is realistic. First of all, in the scientific community, in the US, what's called in the paper title super Mendelian inheritance, which is another way of describing gene drive, has been shown to work in mice, experimental mice. And more recently, the New South Wales government in Australia have just allocated a sum of money to develop gene drive for controlling rodent pests, which are currently having such a high profile in that island south of the equator. What is gene drive? Well, for those of you remembering back to your biology at school, or indeed if you've taken that forward into higher education, you'll remember a certain monk called Mendel and the inheritance that he showed us. And basically, we, as we reproduce, we, male and female, contribute to the genetics of the offspring, the father contributing half the genetics and the mother contributing half the genetics. Each of us carries those two copies through life until we reproduce, in which case we pass on one or other versions from our genetic makeup. What this means is that offspring have a 1 in 4 chance of carrying any specific genotype. 1 in 2 chance it comes from dad, and 1 in 2 chance it comes from mum, giving us a 1 in 4 chance. And on the left-hand side of this slide, you'll see in the blue mouse how that would transmit through a population if there was no selection pressure attached to it. Very low penetrance of in this case, the blue trait. Gene drive is different. Gene drive means that the parent who has the gene drive passes it on to all its offspring. And it does so in a manner that its offspring will then pass it on in a greater number to its offspring until the full population carries the gene drive. It drives the genetic change through the population, as shown in the right-hand side of this diagram. So how is that done? A little bit of the sort of molecular genetics behind this. And in this diagram, I'll illustrate that. We have individuals, whether you're male or female, you have 2 copies of a gene as shown here on the left. We create a gene drive in one of those copies. Its ability, it encodes for CRISPR, it encodes for the gene drive itself, and that copies itself, as shown here, into the other chromosome. This means that every individual carries 2 copies of the gene drive. And as I said, when that is then combined with a wild type through mating, all offspring carry the gene drive, and the process repeats itself, where the gene drive copies itself into the other chromosome, and indeed all the offspring will carry the gene drive. Very new technology, very new strategy, currently being developed primarily in the insect community in a way of controlling population numbers of mosquitoes with the aim of reducing disease transmission from mosquitoes to ourselves. But we can drive any trait through any sexually reproducing species. And the gene drive itself is specific for that species. It's sequence specific. So you could actually design a gene drive for subpopulations or subgroups within a species that carried a very specific genetic makeup. You can drive any trait through the population. Disease resistance, fertility are the 2 that are attracting most interest at the minute, with the latter one, the trait that we've had discussions with ESI about. The concept being that we drive female infertility through gray squirrels, leading to a population decline. We see this as a robust approach to control gray squirrel numbers. We see this as a humane approach. There's no poisoning, no trapping, no killing. The animals exist in the forests and reproduce as normal, but offspring carry female infertility. To demonstrate the impact of this, we have started some modeling to show what would happen if you released gene drive gray squirrels into a population. The population size we looked at in this initial modeling study was either 3,000 or 30,000, and we assumed we were releasing 100 gene drive squirrels. We also assumed that the breeding cycle of the gray was 1 year. You can see in this graph here that regardless of the starting population size and whether there was randomly mating or regional mating, there was a decline in numbers to that of 0 on a different time scale, with that time scale of about 20 years perhaps being optimal. I would stress that this is our initial modeling, and there are a number of other parameters that we could bring in to play here to give us a feel for what would happen. But even in this simple scenario, within a 15-20 year window, you would see a dramatic decline in the grey squirrel numbers. Because this is a genetic approach, you have the ability to lock this into the population for that effect to happen. We see this as a platform technology. As I said, it's being developed within the insect community for disease, to try and prevent disease transmission from mosquitoes to humans. We see this as a technology that could be applied across a number of species, and I indicate a few in the bottom here. The rodents which are attracting the attention in Australia, whether that be mice or indeed in some of our cities, perhaps we might want to tend towards rats. But there's also pests and invasive species in our fields and rivers, including the signal crayfish as options. Any animal that goes through a cycle of sexual reproduction, gene drive could be targeted at. Obviously, animals that have a shorter reproductive cycle, you would see the effect quicker. The concept is the same regardless of that reproductive cycle. I will end my presentation here and pass you back to Andrew Kendall.
Bruce, thank you very much indeed for that. That was very helpful and informative. So if anybody has any questions at all or would like more information about what Bruce has just spoken about, please do get in touch with the European Squirrel Initiative, and we will do what we can to answer any questions you have and help you further. Thank you very much indeed.